Hi, this is Janie, and I'm going to present to you my Probscene 4C Microbial Applications Microbial Fuel Cell. Okay, this is a sketch of my original final design. And I'm not going to go into a lot of detail here, but I built this prototype. And this is a picture of that prototype showing I took a reading right when I started it up, and it gave me 0.207 volts, which was very encouraging. Sadly, the next morning when I woke up, came into the kitchen, there was a leak here, and all of this water was all over the countertop and the floor. So, not trying to be too disheartened, I really like this project and I want to share it with my students before the end of the school year. So I went back to the drawing board and came up with a new design. And this time the salt bridge would enter both chambers from the top, which would alleviate the potential for that leak. This is a sketch of the new and improved design, and you can see that the salt bridge, the hole for the salt bridge to enter the chambers is above the water level in each chamber, so it alleviates that potential for leak there. Knowing that it would take more than one cell to illuminate or have enough power for a small LED light, I decided I was going to build 4 different cells and experiment a little bit with the material used to make the electrodes. So the first 2 cells I found this at Lowe's. It's actually a furnace filter, carbon fiber, a little spongy, kind of porous, and I use that in cells 1 and 2. In cell 3, this is a piece of carbon fiber that the chemistry teacher gave me and told me to try. She thought this would work. They use this in building of airplane wings. And then the 4th cell, I salvaged the electrodes from the first design, and I'm going to put those in the 4th cell. This is the container that I use for both the anode and the cathode chambers. It's just a Hawaiian Punch fruit drink bottle, and this is an example of the cathode chamber. I put C's on the cathode and A's on the anode so I wouldn't get confused. For the cathode, I put a hole in the side right here for the salt bridge to go into, and a hole on the other side about the same height for the air tubing to go into to ventilate it, and then drill a small hole in the cap for the electrode wire to come up through to hold it in place. So all the chambers look very similar to this one. And also the salt bridges are complete. To do these, I just covered one end of the tubing with plastic, leaving a little bit of an air gap, filled it up with a pipette, and then tightly wrapped both ends with plastic, put it in the refrigerator overnight, and it's set. And so these are ready to be assembled too. So I have everything that I need to go ahead and assemble all 4 cells, and then I'm going to go collect my sample. Okay, so this is the last air tube to attach, and then all 4 cells are assembled with the salt bridges and salt water inside of these. And one thing that I did have to do is purchase some of these aquarium T-way connector valves because I only had one pump, and then what I did is I took one hose and teed it off 3 times to give me 4 outlets of air. And so everything's ready, and I'm going to get my sample tomorrow and start them up. When you're ready to go collect your sample, wastewater, you need to take a bucket, shovel, a cup that you can use to get stream water out with. And I'm taking this little spade in case I need to dig into the soil or something, and probably should take boots. Okay, for my sample I chose this creek. This is a creek that flows into the Clinch River. I was looking for a place where there was a lot of debris and wood decaying, and this looks like the perfect spot right there. And there's a little bit of running water across there, so What I did forget to mention is that you need to make sure that your child fits into your boots. Okay, here it is. All 4 cells ready to go. My wastewater sample, I put them in the anode chamber, and they're still really muddy water. I'm ready to start. Turn on the air pump here and get everything running, and then in about 10 minutes I'm going to take my first reading. Using a digital multimeter set on DC setting at 2, the voltage readings were taken initially at 10 minutes after I turned on the air pump. Additional readings were taken and recorded on each cell at 2 and then 4-hour intervals for the first day. And after that, readings were taken about every 10 hours for 6 days. The data collected was put onto an Excel spreadsheet along with observations and any changes that I made to the system. This graph shows the power generated by each cell at each data collection point. Cell 1 performed more closely to what I had anticipated. Over time, the water in the anode chamber cleared up and the voltage reading steadily increased, and then it leveled off at about 0.23 volts. All 3 of the other cells eventually reached around 0.2 to 0.22 volts as well. Before those other 3 cells began producing that amount of voltage, there were some problems encountered and corrected. At least I think they were corrected. One of the problems was with the salt bridges. In 2 of the cells, air pockets developed inside the bridge. This picture shows what that air pocket looked like. And this could be due because either I didn't use enough agar nutrient when preparing the bridges, so they weren't solid enough, or possibly it was due to how small of the tube that I used. Fortunately, the pockets were at the end of the salt bridge, and to fix it I just pulled the salt bridge out and snipped off that portion of the salt bridge that had the air pocket in it. A second problem that I encountered was that the one air pump going into 4 chambers, that one air pump didn't produce enough air pressure to adequately aerate these chambers. So what I had to do, I eventually bought another pump and just divided it so one air pump supplied 2 chambers, and this worked a lot better. The fact that all 4 cells produced about the same amount of power, and they leveled off at about 0.2 to 0.22 volts, could mean a couple of things. One, that the sample was only good enough to generate that amount of power and was a limiting factor, or possibly just that the system size and configuration was the limiting factor. No real difference in the carbon material used could be noted. I felt overall this project was a really good project, and I have already started my senior class working on it, and they're doing their research now and they're loving it. They got lots of questions and are really excited about doing this.